Good afternoon. It's me again. Ready to learn? Yes. All right. Fresh fossils from 4 continents. And if only the word continent started with the letter F, we could alliterate this whole title. But alas, most of the words, but not all. Why are we talking about fresh fossils? When I first saw this, I just thought, there's no way that can actually be from the Tyrannosaurus rex leg bone that you guys are saying you got this out of. But it was published in the journal Science in 2005. I first saw it in 2006 because, I'm not on top of all the journals all the time. But I was close enough to go, what's going on here? Because it's red, and they said this is hemoglobin. That's what's making it colored red. It's the hemoglobin protein. And I knew that it can't last. How long can it last? And then the arrows on the bottom left are pointing to connective tissue. Now these are all liberated from the bone after they dissolve the mineral part of the bone away. Using special solution, anatomists have been using it for 100 years, and it dissolves the mineral part, which is called bioapatite. Bioapatite, it's like little tiny crystals that your bone cells manufacture and wrap protein called collagen. It's a stringy, ropey protein that holds these bioapatite crystals in place, and that's what makes bone flexible yet stiff and also lightweight. It's a fantastic material. So then I thought, how long can these tissues last? I mean, the arrows in the bottom left pointing to connective tissue, and it was still pliable, flexible, elastic, and it snapped back to shape. And so it created this mini controversy that became a larger controversy within the world of paleontology. Paleontology, looking at really old bones and fossils. And so I began to investigate and think and try to look and find out what's going on here. And so here's a conceptualization of what I found. On the top, you remember those little spheres I was telling you about yesterday? And each one, thank you, each one represents an atom. Of course, these are now colored not according to the atom, but colored according to one of the 3 strands, each of which is individually manufactured by these bone cells called osteocytes. And they'll manufacture 1 string, 2 string, a 3rd string, and they snap into position with making a cord of 3 strands, which, as we know, is not easily broken. So this 3-stranded cord, this is just a segment of it. It's actually quite a bit longer in our actual bones, and it's called collagen. Collagen. Have you heard of collagen? Yeah? Oh, good. Well, you already know what I'm talking about, so I guess I'm done. All right. Collagen decays over time, and listen, the decay is relentless. It's unstoppable because chemistry happens. And so you're going to have oxygen react with the exposed atoms on this collagen molecule. So you have nitrogens, you have oxygens, you have double bonds exposed, and so those are going to do chemistry. So you have oxygen will react, water will react, little hydroxyls, and it'll make dust. And the dust will turn into gas. So it should be carbon dioxide and ammonia gas and not much else after just 1 million years or so. And yet we find it, and we found it in dinosaur and other proteins, showing that it must be fewer than 1 million years old. So you can see I'm interested in this because the Bible presents a really radically different world history than that which the world insists upon. The world insists upon billions of years of Earth history, millions and hundreds of millions of years of evolution from fish through the zoo all the way to you. And that's the worldview that our world holds, which is why we're here at this worldview, to learn a different way to think. And our way to think has a different model. It starts with creation. And I think of it this way. From Adam to, let's say, to Abraham, through Abram, is about 2,000 years, if you add up the time markers that are in Scripture. And then from Abram to the last Adam, Jesus, that's another 2,000 years. And the third, roughly 2,000 years, from the time of Christ Jesus to today. So add them up, and it's about 6,000 years. Last time I did my math, 2 plus 2 plus 2. And so that means, so where in this timeline that the Bible presents to us, where in this timeline does the flood fit? Well, if you go by by those markers, especially Genesis 5, you get 1,656 years after creation. You get this flood, which is about 4,500 years ago. So it's several hundred years before Abram. We had this flood. And so that means if we find fossils, and these fossils have original biomaterials like collagen, connective tissue, even maybe hemoglobin in them, then they look a lot younger than what these decay rate studies show. So we can actually measure how long it takes for collagen to turn into dust. And we do that using repeatable lab bench experiments. And as such, each collagen molecule is kind of like an egg timer. And after 1 hour, this timer sounds like what? Yeah, it just goes ding. And so all the collagen in all the fossils in all the world should have gone ding millions of years ago. No, they don't last that long. What kind of egg timers do you guys use in this part of the country? You just like to hear yourself sing. I understand. You sing really well, so I don't blame you. Well, we've done the decay rate study, so we have an energy of activation, otherwise called activation energy, of 173 kilojoules per mole. I knew I would be here to bless you with these facts. Just be blessed. It's a first-order reaction, temperature-dependent, so that means the hotter it is, the faster this will decay. Why? Because it's a degradative set of chemical reactions. It's degrading. So in other words, the hotter it is, the more often those oxygen molecules will bump into the collagen and do chemistry. The more often the water molecules and hydroxyls will bump in and do more chemistry. So the colder it is, the longer it will last. But I mean, how cold do you have to get this so that it will last even a million years? And I mean, maybe even liquid nitrogen wouldn't be cold enough. Of course, that's not a reasonable Earth surface temperature. So you see, we're using the decay rate that we've measured repeatedly, and this is under ideal conditions here. We're using that to estimate how long can collagen last. Because if we can find out how long it can last, right? And if it can't last a million years, which is what we find, and then if we do find it in the fossils, we can use that fact to discern between these two majorly different worldviews, different histories of the world. Does this fit the flood? I think you'll find that it will, at least on the 4 continents that we have time to look at. So how do we measure this? Well, we've done it, but we've followed on the studies that other scientists have done, especially in the world of archaeology. So some scientists publishing in the journal whatever that journal is, Archaeological and Anthropological Sciences, back in 2009. You see this curve, this sort of curve shape? So that's actually a representation of the integrity of the collagen molecule. Is on the left, it's pretty groovy integrity. That's what I'm talking about right there. Every Christian should have groovy integrity, like their freshly made collagen molecules. But when you die, those collagen molecules, they lose their integrity, and they do chemistry, and they break up into pieces, and then smaller pieces, and then chemistry happens. And so at day 8, those little letters D across the top are days. So this is at high temperature. So what we do is we elevate the temperature in order to accelerate the decay so that we can get this experiment done within a reasonable time. We've done it a long 4-month version. We've done a 6-month version, a 2-month version, different temperatures to replicate this. So all that to say, this is kind of how we estimate how long collagen can last under ideal conditions. Most conditions you add to the system are going to diminish the ideal. The biggest condition would be add bacteria. If you add bacteria to the system, is that going to help collagen last longer? No, because bacteria will think it's pretty yummy. Here I am some years ago with my late great colleague, Dr. Kevin Anderson, at one of our labs. We have 3 water baths behind him, and this is how we do this experiment. So we have 3 different temperatures, same bone fragments, and then we measure the integrity of the collagen molecules. We did that, and we published our results in the journal Applied Spectroscopy. in 2023, this is one of the figures that was in our paper. Doesn't this bless your soul as well? Squiggles and modern art. We love it. So within those squiggles, these are actually spectra. So we use a special spectroscope to measure the integrity of the collagen molecule. And then we're the first ones to do that. The modern art is actually a way to use a special kind of microscope to visualize collagen. And in the second column, let's just point this out, just so you have a clue of what I'm talking about, just a little bit here. We have the red bits. That indicates collagen is present. So when this laser shines on the bone, collagen and only collagen will resonate with that particular wavelength, 920 nanometers, and it will be detected on the camera. And then, so with time, at high temperature now, the collagen diminishes in little bits and spots, and then now we're down after day, was that say, 18 or so? We've got very little to no collagen left, both visually and spectroscopically. Okay, so what we're saying is we're using a new technique to do the same experiments that they used to do to determine what? To determine with greater accuracy than any scientist had done before, how long can collagen last? And what did we confirm? We confirmed the results that they already obtained. Collagen cannot last 1,000,000 years. But don't take my word for it because I'm biased, because I believe the Bible. As though an anti-Bible bias is not a bias. Yes, it is a bias. So you can't avoid bias. So what do? You just pick the best bias with which to be biased. So analyze your biases. That's what we're here doing at a Christian Worldview conference. They said this, publishing in Antiqua back in 2011, extrapolation from high-temperature experimental decomposition rates, which is what we just did as well, using this activation energy, which is an experimentally determined value, suggests that at constant 10 degrees Celsius, collagen can last between 200,000 and 700,000 years. Wow. Hundreds of thousands of years under ideal conditions, collagen can last a long time, at least bone collagen. You know, we have an analogy for this. Anybody heard, raise your hand if you've heard of the Dead Sea Scrolls? Dead Sea Scrolls, yeah. So most of the scrolls in the Dead Sea collection, the caves of Qumran, are what? Made of parchment. And parchment is made of sheepskin. And skin of any animal is 97% what molecule? Collagen, yeah. So, and it's 2,000 years old. And so, but what does it look like? The edges of these parchments look, okay, let's do the hand thing. The edges look, yeah, in the back. In the back, yes. Pick one. Gray. They can't decide. Do you want to sit? Yeah, go ahead. Oh, well, you're doing this. So I thought we were communicating, but apparently. So what do these Dead Sea Scrolls look like? You've seen them, pictures of them, and they look like what? Do they look fresh as a spring breeze and a basket of fruit? No. They look burnt. Okay, so what else? Yes. Frayed around the edges? Yes. What makes them look old? Yes. Petrified? No, not petrified, actually. Yeah. Right behind you? Yellow? Yeah, they get yellow and then darker and then brown on the edges. So that's a result of oxidation reaction, the very reactions that take collagen and turn it into dust. So first it goes yellow, then it goes brown, then it turns into frayed edges, and then it frays. And that's why they have the Dead Sea Scrolls in special cases that are temperature controlled, UV light controlled, and things like that, to help them not decay quite as fast as they otherwise would. All right, you got the idea? Collagen should not be in a bone that's older than 1,000,000 years, just 1,000,000. And yet my colleagues insist that all these fossils come from all these layers, were deposited tens or even hundreds of millions of years ago. They've got a question to answer, and that is, why do we find collagen? I can give you the answers they typically give, but we're going to just survey some of these results that the scientists have come up with. As of last 2 weeks ago, we're up to 131 different technical journal articles that describe original biomaterials like organics, and even some whole tissues like blood vessels inside fossil bones. Let's go to the first continent, Asia. Shall we fly to Asia together? Whee! We're going to hit our first dinosaur and first fossil. And go ahead and say Psittacosaurus. Isn't that great? It starts with the letter P. Love that. So this is in China and the Gobi Desert. So some of the first really well-preserved fossils were found there. And so you can see this one's actually still in situ, which is short for in situation. So you can tell it was splat, buried in mud. And I think he died in the flood. That's what I think. Why do I think that? Well, one reason I think that is because, A, animals don't get preserved like this anywhere today, but they got preserved like this everywhere in the past. And what does the Bible say about the flood? It happened everywhere in the past, but nowhere today. No wonder fossils aren't forming today. We don't have the flood happening anymore today. God is true to his promise. He said, I'll set my rainbow in the sky as a sign of this promise. I will never flood the earth again. He hasn't. We don't have fossils happening today. I think that's connected. But on the right, you have these pretty colors. And this is from a special microscope called a cross-polarized light microscope. And what it does is it detects collagen fibers that are still intact enough to twist the light as it goes between the two polarizers. You use polarized sunglasses? You ever done that? Use polarized? Have you ever taken like two polarized sunglasses, two pairs, and then you turn one 90 degrees to the other and it turns black? Yeah, because the one polarizer stops the light waves that are going in this direction. Yes, we could do the wave. And then, so now you've got only the light going in this direction, and then you take the other polarizer, which stops all the light going sideways, which is harder to do. And then by now you've got all the light stopped going this way, all the light stopped going that way, and you got no light going through. So it blacks it out. It's called extinction. And so you adjust the polarizers to extinction, but then if you put something in between those two lenses, something that will twist the light so it will make it through the next polarizer, then that's what you have here. And it turns out in fresh tissue, in fresh thinly polished bone slices, called thin sections, or in fresh skin even, it will twist the light so it makes it through the polarizer, and it makes these beautiful colors, gold turning into blue as you rotate the sample on the stage of the microscope. Anyway, they published this in the Proceedings of the Royal Society B. But about Nature, the world's most prominent scientific journal. They looked at the embryology of an early Jurassic, we're talking, what, 150 million some odd supposed years? I don't think they're real years. I think they're Monopoly numbers. Here, have some years. I don't buy it because of this. From China, with evidence of preserved organic remains. A subsequent study on the same sample found that it was collagen protein. So here's the picture, here's the story. And by the way, if you want to say it, it's a fun name. It's Lufengosaurus. Lufengosaurus. It found near Lufeng, which is a city in south-central China. And there they found so Lufengosaurus is like it's a sauropod, so long neck, long tail, 4 feet on the floor. Anyway, they found an egg, and then lo and behold, this egg had the embryo still in it. So they sectioned it. They destroyed the fossil. Yes, you can always glue it right back, so relax. And then they found the little baby had a femur, that's the biggest bone in its body, and then they sectioned the femur, and they put that under the microscope and did some other analyses as well. And this is what they found. They found, and this is their quote from their paper, thus it can be concluded that complex proteins, i.e. collagen, were preserved in our specimen. That's interesting choice of verb. They were preserved for 100 millions years. It's like, here comes the magic wand again. What about the chemistry that shows that they can't preserve even 1 million years? They avoided that whole issue by just saying they were preserved. And I think they are there. I think they're doing good descriptive science on this one. Okay, here's a turtle called, everybody say, Mongolemys. And they found these bone cells liberated from the bone tissue from this turtle fossil. Not just dinos, folks. It's all kinds of critters buried in fossils. And these osteocytes still have, some of them still have a little dark circle that stains when you put DNA stain on it, and it looks like the nucleus is still inside these cells somehow. I think these are thousands of years old from the flood, not 1 million years old. They even have these dendrites, these branch-like projections that come. So that's how your bones are being manufactured right now. And you lucky young folks, you get fresh bone happening all the time. And the older we get, our bones get more and more brittle because these osteocytes, they slow down. And so they're like, yeah, I'll get to making more collagen tomorrow. I'm kind of tired today. And so that's why our old people's bones break easier and heal slower. Which tells us what? Even in our own lifetime, collagen doesn't hold its integrity. So chemistry is happening, destructive chemistry is happening even in our own bodies. These are the cells responsible for making new bone, and so they've been liberated from the turtle fossil. Okay, here's another fossil. Everybody say Caudipteryx. Caudipteryx. Now, many evolutionists, and honestly, some of my creation colleagues will say, these are feathered dinosaurs. So I put feathered dinosaurs in quotes. You can go with that. I'm not going to fight you on that. I'm just saying I'm not convinced that this is a feathered dinosaur because there are evolutionists who look at this same fossil and they say it's what they call a secondarily flightless bird. They say it's a bird and not a dinosaur at all. It didn't have wings big enough to fly, but you know there are birds today that don't have wings big enough to fly. That's okay. Have you heard of the flightless cormorant? It's on some of you have. That's because you're homeschooled and you know stuff. Yes. Great audience. So it's got these little wings. It doesn't need to fly. It's on this island. It has everything it needs on this island. And so it lives its groovy life having deployed a version of itself that has little tiny wings, enough to maybe swim, dive, but not enough to fly. And maybe that's a variety that God made of the Caudipteryx as well. Well, who cares? What about the Caudipteryx? Has to do with soft tissue. Well, publishing way back in 2021, scientists published in Communications Biology their discovery of chromosomes in Caudipteryx. Now, these birds were buried in the same layers as dinosaurs, discovered in China, described in this journal. It's on our list of 131 now. And there you can see on the right, top right with the pink background, it's stained pink. These little circles are chromosomes, so it took up so the DNA must be in there because it took up the DNA stain to show whole chromosomes still in there. And these are in cells. These are cells that are called chondrocytes. Chondrocytes make cartilage. They manufacture cartilage. So they found this in the joint, the knuckle joint of this bird, still intact there. Okay, so that's the first continent. Guess what? It looks like all these fossils are showing in all these different rock layers, different places around China. China's huge, by the way. It looks like over here it was deposited recently. Over there it was deposited recently. Looks like Asia deposited recently. Recent deposition of fossils across Asia. And we get to fly way over to Europe. Here we go. And in Europe, we're going to see something similar here in Belgium. Everybody say mosasaur. Mosasaur. Not a dinosaur, but it's an extinct marine reptile. Used to swim in the seas. And I'm glad it's extinct because it had a huge mouth with lots of teeth. And I wouldn't want to go swimming when this thing was alive. But these images, I know they're kind of weird looking to the uninitiated, but trust me, this is really scientific. Let's just outline some of this. So this is a special microscopical technique showing an osteocyte, remember, with the dendrites, those projections that go out into the bone. So they dissolved away the mineral part of the bone, and they're like looking for tissue. Is there something soft, organic, biological that's still in here? And there it is. And it's not a bacteria, because bacteria don't look like this at all. And then this is my favorite one, in the center, letter H, because this is just loose, floppy tissue that was liberated from the bone, and it's after they dissolve it, it's just floating in the solution, and then they just put that on a microscope slide, shine some light through it, and a high school a student could do this very experiment and just take a picture of what's called ECM, or extracellular matrix. It's made up of collagen fibers interwoven to make up the fabric of bone right there from Belgium. Okay, what else? Let's go to Poland. And over in Poland, we have this is SEM, I think, electron microscopy. Now, on the top left here, We have bone here, but then there's this tube sticking out of the bone. You see this tube? It's sort of projecting out toward you. Or does it look like modern art again? Hopefully you see this is a tube, the dark interior, and then inside another tube, they found these little bitty spheres, little disc-shaped structures. And what did they call? By the way, this was published way back in 1998. We should have been paying attention to this way back then. But it was black and white images, okay? And it wasn't until 2005 that we saw picture images, and you know, picture tells 1,000 words. So it was those color images that really got the world's attention. But they were saying it even back then. Erythrocytes, which being translated means red blood cells. Red blood cells, that's what they called it, have been present in dinosaur hemoglobin. And this is called everybody say Tarbosaurus? Tarbosaurus. It's like a T-Rex, except smaller, because of the theropod dinosaurs, which country has the largest ones? Poland or USA? So T-Rex, bigger than Tarbosaurus. But varieties of the same created kind, in my view. Let's go now to Poland. Hey, same place. And they found a creature called Nothosaurus, but this was actually in layers quite below the Tarbosaurus. And these are called Triassic in the mainstream system. I think these are middle of the flood layers. So thinking about the flood, we've done a lot of that, and a lot of more than just thinking. We've mapped the results of the flood around the world using 30,000 different drill core boreholes, and looking at the thickness of the rocks and the rock type, and we matched them across the boreholes. And what we found is that there's shallow marine fossils in the lower ones, deep marine creatures, shallow marine creatures, more shallow marine creatures, then wetlands, and then swampy creatures, and then at the very topmost layers, and there's not very many of them, we have some hard ground living creatures like camels. And that's, by the way, where I would look for the pre-flood people. I'm not sure that we've found them yet, but there's not a lot of places to look for them, and there's not a lot of places that will let a creation person look for them as well. So anyway, so that's, and I think this is not a representation of, hey, we evolved from seafloor creatures that evolved into shallow marine creatures, which evolved into fish, which evolved through the zoo all the way to you. Like they think this is a record of evolution, but that's just imposed upon the data, not from the data. Instead, we see this as a record of the progressive floodwaters as they went higher and higher, and finally up onto the edges of the pre-flood world. I think it was a single continent, something like Pangaea, that was broken up during the flood, broken up by the flood, into the continents that we have today. And then finally the floodwaters, after day 150, says the scripture, the floodwaters were what? Over the tops of the high hills that existed in that world. Okay, anyway, so we have blood vessels from Nothosaurus, which means all these layers. It doesn't represent periods and eras and everything like that. It just represents earlier in the flood and later in the flood. But you might have thousands of feet of sedimentation happening because of this flood. You know what Peter said in 2 Peter 3, verse 6? The world that then existed perished, being flooded with water. And I think this is the result of that, because we see this worldwide. We're seeing it in Asia, and now this recent sedimentation burying these creatures that couldn't get out. Anyway, Nothosaurus looked really cool. I'm going to show you a picture of Nothosaurus Wednesday. Does that mean tomorrow? Yes, tomorrow. So you'll remember Nothosaurus, and we'll look at that tomorrow. Now this is stunning. This is a worm casing, a seafloor worm, and it's called, if you want to say it, Sabellites. And they got this from a drill core, deep sea core from Russia, and beneath the sea there's sediment, and then that sediment is from, they found some sediment that they labeled Cambrian. We have Cambrian sediments in lots of different places. Around every continent has some Cambrian sediment. Not everywhere on every continent, but every continent has some representation of it, I think. And this, generally speaking, is the first or lowest sediment, first to be deposited, I think, by the flood, that has any fossils in it at all. The conventional age assignment is 500 million years, sometimes 520 million if you get to the bottom of the Cambrian. Again, I think those are monopoly numbers. Why? Because they found chitin structural protein composite. Proteins lasting 500 million years when they can only last 1 million. They can't even last 1 million based on decay rate studies. And what does the flood model say? The flood model says that layer down there, this layer, all the layers in between, and the layer on top, all in 1 year, just 4,500 years ago. No wonder we find proteins here, here, down here, and at the very bottom where we find the first fossils. Wow. This really fits, starting to fit the flood model quite well. And those, you see a picture, those of these fibers that make up the worm casing. It's like a worm home. And so this particular sea worm, he, if you can imagine like a tube, and he secretes the tube, and it's made of chitin, which is like what an insect body is made of, except this is a mixture of chitin fibers and structural protein fibers mixed in it to make a tube. And then what does a worm do? He extends a fan when times are good, but then when he sees a shadow coming overhead, what does he do? He pops down into his home. And by the way, the Sabellides worm casing looks identical to Sabellides worms that are still alive today, showing that no evolution happened to this type of seafloor worm in a supposed time span of 500 million imaginary years. Let's go now, zooming on a plane to South America. I think the front row gave us a crash landing. Don't do that. In South America, we see, everybody say, Titanosaurus. One of the largest land creatures to ever have walked the earth. Yes, it was monstrous. Not the longest, but maybe one of the most massive. Anyway, even those biggies, the big giant ones, 130 feet long, some of these, even those came from eggs, hatched out of eggs, the size not much bigger than a football. So they all started pretty small. And they probably laid a lot of eggs. And as they were walking around in the floodwaters, sometimes swimming, sometimes wading, how do I know? Because I've seen the footprints. I've seen the footprints in a lot of different places and seen pictures of them in other places where you have full tracks in the mud that got preserved, I think, during the flood. But then you see scrape marks where their bodies were suspended by water that was at a certain depth, and their feet couldn't quite reach the bottom, just enough to make these little toe scrapes as they were struggling in the rising and falling floodwaters. Genesis 8:1, and the waters, holuk vashub, went back and forth. King James translates it assuaged. The waters assuaged off the surface of the earth. So for months and months, these floodwaters are sloshing back and forth, and I think these critters were struggling to stay alive. And what mama is going to want to hold on to her eggs if she's just not even having a good day or a good month or a good year? So we're waiting for these extremely tough creatures to die off. And finally, the last creature that was not on board the ark, not any creature, but what was on the ark? Did God send fish to go on the ark? No. He sent any creature that had the breath of life in its nostrils. So that word is in Genesis chapter 7. And so these creatures had nostrils, and we can see that from their anatomy. Anyway, they found ovalbumin. There it is, ovalbumin. They detected and published this in 2005 from this eggshell. Ovalbumin is an egg protein that is today. So you're saying that there's egg protein in a titanosaurid egg? I'm not saying it. My secular colleagues are saying it. And ovalbumin shouldn't last any longer than collagen. In fact, it should last a lot shorter time than collagen. Same is true with DNA. It doesn't last as long as collagen, which is why we focus on collagen, like this study did, published in Langmuir. So there's modern bird collagen, which shines brightly green using this particular technique, and then the extinct bird collagen also shows up green. I don't remember what the circles are supposed to be highlighting. It's been a minute since I read this paper. But we see this is a different technique used to illustrate or to image the same phenomenon. We use this to look at collagen, and guess what? The secular community says that can't be collagen because we know, based on biochemistry, that collagen can't last even 1 million years, and we also know that that's 70 million years old, so whatever you're looking at is not collagen. Well, we think it's collagen because it passed this test and that test and this test and that test. And here's yet another test that says the same thing. Yes, collagen. Okay, the fourth and final continent, we get to fly north. Here we go. North America. Landing so smoothly. We land in Wyoming. And here we are at the Green River Formation, famous for its fish fossils. A lot of fish. By far the most abundant fossil vertebrate is fish. Our fish is fish. Anyway, also there's half a lizard, the back half, in this same shale matrix there. And so these scientists publishing there in 2011, they zoomed in on what makes this look dark. Okay, you see the question? Why is it that the animal remain dark? I mean, if this is billions of years old, millions of years old, if it's eons old, then all the original protein should be gone, long gone. So what's making it dark? Did the animal attract some mineral that stuck to the animal, and that's why it's dark? What is going on here? Then they zoomed in, and then they zoomed in further, and they found that each of those little dark circles, these little dark circles here, dink, 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 they look like scales, because they're reptile skin scales with the protein still in there. And they found keratin protein. So some reptile scales, even today, are made of keratin. You know, keratin's in your hair, and it's what makes up your fingernails. Keratin protein. It's tough, right? But it doesn't last forever. And so what these scales, some reptile scales have, is like a little bitty micro fingernail worth of keratin, but it's wrapped in skin to make them have tough skin. And so this fossil creature, lizard, had the same thing, and they found the keratin still in the skin. So they said minerals came in and locked the keratin down, and that's why the keratin hasn't done any chemistry for 40 million years. I think it's a story, and in fact, the story has flaws, because what's going to bring minerals in place? In your imaginary tale about minerals being brought in, water. And what does water do? It reacts with the very chemicals you're trying to explain, preserved. Well, I'm going to narrate this next video, if it'll show, because I don't think I have audio. Will it show? No, it won't. Yeah. So this is Dr. Schweitzer. This is 60 Minutes news program. And she's the leader in my field, and she's the one who published that 2008 article with T-Rex tissue. And these are the results of demineralizing bone, and she's showing Leslie Stahl, the host, what she found in the microscope. And Leslie Stahl is saying, wow, that looks like it's fresh tissue, like it could have been, from yesterday's bone. But this is 70 million years old, so what do you think? And then Dr. Schweitzer is on the right, and she's saying, well, I asked my lab technician to do it again. So he did it 17 times, and he kept pulling out these flexible tissues. And then what are we talking about here? We're just shocked. So that's kind of what we're talking about. And so the scientific community said, that can't be from T-Rex. It has to be, we know that can't be from your T-Rex. You must have dropped your lunch in the test tube during break time. So Dr. Schweitzer got a different lab, different set of co-authors and experts, went to a different country, a different rock layer to get a different animal. And she got Brachylophosaurus, which is a hadrosaur. Just say hadrosaur instead. Yeah. So they got a hadrosaur. And guess what they found? Again, blood vessels again in this hadrosaur as well, this time from Canada, eh? And even with the little bitty red discs that are inside the blood vessels, which they called red blood cell-like elements, maybe they're red blood cells that are left over from the original animal. There's evidence of that in many other samples as well. So branching blood vessels, other experts at the time said that can't be from your hadrosaur. It must be bacteria that are recently manufactured this stuff. But bacteria don't make branching tubular structures with red blood cell-like elements in them. So that's been refuted. And and they sequenced collagen. That's the gold standard. What is the protein sequence of amino acids that make up collagen? And they found that sequence in T-Rex, hadrosaur, and our group just published our collagen sequence from Edmontosaurus. We just got it published in the journal, Analytical Chemistry, in January. So 2025 is a great year to be a Bible-believing Christian, because if we believe our Bibles straightforwardly, we would conclude that the flood is a real event. It was global, according to the Scripture, worldwide, and recent, according to the Scripture. And we see evidence that supports exactly that from all these 4 continents and others. Triceratops tissue there on the left, more ECM, extracellular matrix. And on the right, you see the blood vessels fragment. So they're fragmentary. They don't last long. So once you extract them from the bone, you better take your pictures quick, because they're so degraded that they turn into dust as soon as you expose them to the air in the laboratory within weeks or months. And then on the far right, more of those osteocytes from, everybody say, Triceratops. Tri means 3, so you have 2 brow horns, 1 nose horn. And so the horns are just what we see on the fossils, replicas, and the fossils that we get out of the ground is we see the horn core, which is made of bone. But like today's horns have horn cores as well, which are made of bone, but it's keratin horn that's manufactured by the bone cells, and it wraps around that bone. So today's like cow horns are made of the horn core, but it's keratin that makes the outer part of the core, outer part of the horn. And guess what other researchers have found? Keratin as well from these Triceratops horns. What else? We got another mosasaur, but this one's from Kansas. And the Niobrara Chalk Formation in Kansas, huge. It goes from Canada to Mexico. Because as we all know, sediments get deposited across whole continents all the time today, right? No. So we have this continent-wide dispersal of sediment and creatures mixed in with them, and mosasaur is one of them. This was excavated in 1967, but a really good specimen, something like 25 or so feet long, and they had to break it into sections to transport it to the LA County Museum of Natural History. And I didn't know that till I got there, but I had read this study before I got there. And there they're talking about this little red patch right in its chest cavity, right there, if you could see it. And they said, by the way, we found skin scales made of skin, still just skin, not stone, stuck down onto the bones across this whole naturally mummified carcass. They even found, and you can read the paper, it's free online, PLOS One, they found still colored purple, the retina from the back of its eyeballs there, and they found dried down poo-poo in its guts. And what else? They found the red patch, and they said, why is this patch in its chest red? And guess what they found? Here it is, the presence of hemoglobin decomposition products, the residues of visceral organs. In other words, that's what's left of its heart, and it's still hemoglobin enough to register as hemoglobin with the tests that we can do. So I was there, and I didn't expect it to be there, but when I saw it, man, I was hopping up and down. I said, look, its heart is still red. So there's me pointing to the red heart of the mosasaur. And the other people in the room were like backing away. No, wrong direction. Look, it's bloody. It's over here. You're bloody weird. So there's my photo of the heart residue. That's what they called it, these secular scientists. Okay, so we have hemoglobin, elastin, chromosomes. What else have we covered? Of course, collagen and ovalbumin. Let's go way down here in the Permian layers and look at this creature called Diadectes. Did I say that? Diadectes. So imagine a big, scary salamander, 8 feet long. And I think these are, by the way, this rock column, it's representative, but there's this stack exists nowhere on Earth in this form. Most places have just a few of these layers, maybe a few of these layers, and maybe none of these, or vice versa. But if you were to add them up from over there and over here and over there, then you can stack them together and build this diagram. But it helps to illustrate it helps to map where these things came from. Anyway, I think this is early flood, mid-flood, late flood, and then this one up the top is like Ice Age, so it would have been after the flood. That's it. The rest of this, I think, are flood layers. Why do I think that? Because these layers are so broad and extensive. They go for hundreds of square miles each one. And that doesn't happen today. Today, if you have a flood today, it just fills the river valley, dumps out of the ocean, and it makes a fan shape called a delta. And so these are not delta-shaped. They're not stuck in a river valley. They just cover, they go for hundreds of square miles, and they've got fossils in them. I think these are underwater, tsunami-like pulses during the flood. Flood water's rising, especially in these lowlands. The pre-flood seas, shallow seas, got deposited on. And then finally, the floodwaters here are receding off the earth. And most of these layers up here with the dry ground living creatures are actually offshore. They're under today's oceans. Anyway, Diadectes shouldn't have, with an age assignment of 250 plus million conventional years, shouldn't have any collagen, but a buddy of mine found a Diadectes, and this is his lab. And this is a drawer that he has with all these microscope slides, and those are his microscopes in his lab. You can tell he's a real tidy fellow. Keeps it all, everything in its place. Every place for a thing and everything in its place. Cough, cough, cough. But man, does he do good microscopical work. So this is the Diadectes bone thin section. You can see features of bone. You've got these striations or stripy patterns. You've got these little black dots, those are called lacunae, and that's where the bone cells or the osteocytes, they live in these little gaps called lacunae. You can also see this big black crack letting you know this is fossil bone. And this is under cross-polarized light with the microscope setup. Then you add something called a red filter, and boom, it pops. And you've got these purples and blues, and when you rotate the stage, the blues turn gold and the gold turns blue. That's how you know you've got collagen still in the bone. So we found collagen in the Diadectes. This is a world first discovery, hasn't yet been published, but you guys get to see it because you're attending the Worldview Conference. And in another place, you could see the thin section of the bone has, this is a place where a blood vessel once penetrated the bone, and this is, the blood vessel's gone, I think, but the bone still has gold and blue, showing there's collagen still in parts of the bone. Now, this part where it's purple, I think the collagen's all gone. What else? We have another Edmontosaurus, and this is an image I took, and we were able to rotate the stage, so hopefully it'll work, and you could see the gold bits turning blue and vice versa. This big white is just calcite that has precipitated in the pore space inside the bone. So that comes from outside the fossil, but I think this is actual bone with cracks in it. It's the bone remnants, and watch it turn and shift. There it is, boom, turning it to gold right there, letting us know that parts of this still have collagen in it. And that's one of the techniques that we used on our recent paper in the journal Analytical Chemistry. We have more. There's lots more. Here's Edmontosaurus protein sequence, and here's the list. It's at tinyurl.com for HTM, blah, 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 blah. Or you can go to icr.org and just look up, where's Dr. Thomas's soft tissue list? And we have it on our website as well. But conclusion, bottom line, all 4 continents look freshly deposited, like recently deposited. So we have these little stars marking where we found, this is just some of the discoveries, but if I were to put all 130 on a map, you'd have stars all over the place. Probably I should do that, but this is enough. All 4 continents. You got to have a worldwide cause to produce a worldwide effect. I think the flood, having been worldwide, is a good explanation for a worldwide cause of watery, catastrophic deposition recently, thousands, not millions of years ago. We talk about these things in our Guide to Dinosaurs, which we brought, our Uncovering the Truth About Dinosaurs, TV-ready, highly produced, on location. We do a dino dig there on these discs, and disc number 4 highlights original biomaterials in fossils. And so you can get that. It's great for church studies, and it's also entertaining. The Guide to Dinosaurs is for families, so it's a family book, a little something for kids, and some stuff for mom and dad, and everyone in between. My little book, Dinosaurs in the Bible, I wrote really to answer several specific questions. Is there evidence that dinosaurs lived after the flood? My answer is yes. I show some evidence in this book. It's really a booklet, Dinosaurs in the Bible. And after I talk about dinosaurs, everyone always asks me the same set of questions, one of which is, if the evidence is so powerful and strong that these were deposited in Noah's flood, then why don't all the scientists believe that? And so I point to Jesus' dealings with the Pharisees as a model for why scientists don't just believe everything according to the evidence. And that's in chapter 4. Anyway, CBAB, Creation Basics and Beyond, we've already been talking about. We even have kids' books, so if you have siblings, kids, go ahead and order the book from us, icr.org/store. And that was my last slide. I went right past it. Wow.